me a story in the past that I think really stuck with me, which was when you conceived of, which is now 25 years ago, right? A lot of the early patents in regrowing organs from stem cells are patents that you filed, that you have. But when you first recognized the power of the stem cell as a regenerative tool, could you tell that story?
So, Peter, as you mentioned, I spent the beginning half of my professional medical career as part of a surgical team at Cornell, where we were taking care of head and spinal cord injury patients. And we got really, really good at developing the techniques and the approaches and the technology to manage the acute period after a severe head or spinal cord injury. But we couldn't return these patients back to a normal neurologic functional state. And we always felt that that was the best you could do because we couldn't unlock or uncover the natural repair processes that would fix a brain injury that paralyzed somebody or spinal cord injury that paralyzed someone and allowed them to walk again. And I was always convinced that there was enough plasticity, but there were other tools out there we could use. So I embarked upon this little mission to better understand developmental biology, a subject in anatomy that I kind of avoided and glossed over. And a couple of events took place which led me to think that resident in the materials that were being thrown away at birth, we might find some of the tools that would allow us to reprogram a human neurologic system and get it to return to function.
So what were those? I mean, there were two concrete stories you told me that sort of like got me thinking about this in a very different way.
The first was that when my oldest daughter was in utero, she's 26 now, so that goes back a long way, and I was a young surgeon in the surgical ICU, I went down to the ultrasound suite to look at my unborn child, and for the first time it dawned on me that although she was a peanut-sized embryo, the placenta had already grown into this fairly large organ. When I was in medical school, we were taught that the placenta was the vascular interface between the mother and the fetus. I'm an engineer. If that was true, they would grow at the same rate. So I said to myself, this must be an indication that the placenta was a rate limiter and was actually dictating the rate of growth of that embryo into a fetus, into a newborn. And if that was the case, what was it doing to be that rate limiter? So as the field of stem cells was emerging, and we recognized that we all start out as a single cell that divides and differentiates into all the complex, specialized cells of our body, that there must be a governor for all of that, and I believe that governor resided in that organ that was expelled after birth and thrown away. So maybe the hoarder in me decided I was going to go and look at these placentas and see if I could find some of this material that could be used to reprogram the body.
The second story, which was impressive, was talking about in utero surgery of a fetus.
Exactly. So the other thing is that, you know, as somebody who liked to see what my colleagues were doing, I got to witness fetal surgery, which is, you know, we live in an era today, we lived in an era 25 years ago where you could identify in utero that there was a developmental defect leading to something that might be an irreversible problem in the newborn, something like spina bifida. And we figured out that if you could uncover that early enough, you could actually open the womb with the developing fetus inside, leave it connected to the placenta, surgically repair that abnormality, put it back in the uterus, come back in several months, and when the baby was born, not only was the spina bifida taken care of, but there was no evidence of a scar. And so I saw how gross the surgical repair was. For the baby to be born and there not to be any evidence of the surgery astounded me. And I started saying, hey, is this a way to unlock a human being's inner salamander?
Yeah, I mean, amazing and miraculous, but I totally buy that. It was those two stories that got me saying, oh my God, this is the stem cell. So let's talk about what are stem cells, why are they important, what do we know about them?
So I'm going to stand up because when I sit in this barber chair, I'm worried that you're going to trim me. So, and by the way, this is the first time I've gotten on this stage from up here. And so remarkably, I actually found a miniature bust of Dan Kraft, and I'm going to turn this into a lapel pin. So stem cells are remarkable in that they have the ability to divide and replicate themselves so that you can have twice the amount of versatile, undifferentiated cells, and those can serve as a reservoir that can be used for a variety of purposes during life to replace, renovate, repair the body. And they can also differentiate, that is, mature into the specialized cell types of the body. It's that unique versatility that resides in stem cells that first made them an interesting potential therapeutic tool. So you and I talk about this a lot, Peter. Cellular medicine and stem cells have been around a long time. Why has it taken it so long for these to be reduced to common clinical tools? And so I call this the fog of cellular medicine.
Is this you landing this jet?
Yeah, this is me landing in the fog, and one of my naval aviator buddies out there will recognize this coming in, not seeing the runway till you're 20 feet above it, or the carrier deck. And the reality is that fog is something that has resided in our industry because there was an absence of a deep understanding of the underlying biology of stem cells, and more importantly, there was an absence of understanding what cell was actually going to be the one we derived into a product. Today, after 20 years of investment, we know that we can use stem cells to reprogram the transcriptome, the synthetic repertoire of the cell, and we can also use stem cells as starting material to produce things like immune cells or to produce cells that are components of engineered organs and tissues.
So one of the things, I mean, you've given me this this vision. We'll talk about it a little bit later more, but one of the aspects of why we age has to do with what?
So now I'll sit because we're talking about aging. You know, we actually recognize that all of us, everybody sitting in this audience, the vast majority of the cells in your body and tissue are about 2 years of age, meaning that they got to their differentiated form less than 2 years ago. We are constantly undergoing a process of renovation and remodeling that's driven by the population of stem cells resident in those tissues. And so one of the things we recognize is that if you have these cells and they're constantly renewing you, by getting cells therapeutically, can that serve as kind of a tool to replace, regenerate, restore our health? So the old excitement and optimism around can a stem cell a day keep the doctor away is something that drove me into the field.
And so, I mean, one of the things that you've taught me is that we're actually, our population of stem cells through our body is diminishing over time.
That's right. That's right. So if you think about this, a stem cell is a source of uncorrupted genetic information that can be used to constantly drive the remodeling and reprogramming of your body. Depending upon where you get those stem cells, you may get them at a very, very early state where they haven't accumulated any defects, and they have all of the capabilities of becoming any differentiated specialized cell. Now, that's why some of the challenges in turning these into products was where you get the cells, what the underlying biological attributes of those cells are. Is the cell source scalable and of high enough quality to meet a regulatory standard so the government allows us to use them to treat patients? And underlying all of this is how much will these products cost? From my vantage point, cellular medicine was not going to have an impact, a meaningful impact in healthcare unless we could deliver these treatments in an economical way. They had to have a positive impact on the cost of delivering healthcare.
And so those are the challenges, and the solutions then going forward are what?
So this is an example of fetal surgery. My first sort of the epiphany of saying, hey, if the regenerative potential that resides in utero could be harnessed, that might be the solution to all of these problems because placentas are abundant. The
catchphrase of the day. Yeah.
Catchphrase of the day. In this country alone, 4 million placentas are thrown away every year. You can qualify the material, subject it to rigorous controls so that you're sure and certain that the product meets a high pharmaceutical-like standard. And the unique biology of the placenta is something that provides almost limitless clinical applicability, and we'll talk about that in a second. So, the placenta, here it looks, I love this picture because it looks like this is Starship placenta, doesn't it? It's, and the other nice thing about it is it's an organ that gets disposed of, thrown away, and it comes with that coaxial cable attached to it, which lends itself to all kinds of engineering applications. And remarkably, the placenta is nature's stem cell factory, and it's the richest source of cells and biomaterials for therapeutic purposes.
It's sort of like the 3D printer that produces the baby, so to speak.
That's a term you used, and I love it because, in fact, every placenta produces a human being. Right? It is solely responsible for producing that human being, but also maintaining a safe environment for the development. Remember, in evolution, human beings, pregnant females, were under tremendous physiologic stress, starvation, injury, etc. Humans devote 9 months of reproductive energy to a single offspring. So evolution has selected for a system that protects that developing fetus at all costs. And that life support system and that 3D printer, if you will, is responsible for the perfection that exists at the instant of birth.
One of the things that well, do you want to talk about this slide first?
Well, so the other great solution that the placenta provided insofar as it being a source of cells, for those of you who follow this field, you know that when stem cells first emerged, we thought that we were going to have to match every donor to every recipient. And as a guy who did my rotation in transplantation medicine, that's easier said than done. How often do you just stumble upon the perfect match of donor to recipient?
This is, you took this photograph, and these two people did not know each other. Where were you at a game?
I was at a game, and I saw this from way up in the bleachers, basically, and I had to run down and take a photograph. I said, aha, here it is, the perfect way to describe matching to a donor and a recipient. So, and I don't even think that they even recognize that perfect match.
So Bob, one of the visions you talk about is the notion of the stem cell as a computer.
Yeah. So a couple of things. A lot's changed in the field, and some of the concepts that you and I have come up with over the years on how to explain this tool in a way that everybody could recognize.
So what's changed in the near term?
So number one, we understand the biology of stem cells better today than ever imagined. We understand that what they are is they're a delivery system for an uncorrupted genome. There's pioneering technology in manufacturing them, storing them, and so on.
Let's slow that down 1 second. So one of the things that you taught me is when you're able to preserve a placenta at birth, and right now, LifeBank, the company, so Bob started 5 companies, the last one scaled to over $1 billion. He's been, like I said, my big brother, my hero. You've stored about 70,000 placentas as part of LifeBank. And what you're effectively doing when you do that is you're storing the baby's uncorrupted boot disk of the original DNA of that child before it encounters radiation at 40,000 feet or superoxidants or all kinds of things.
Right. So again, parents, for years have had the opportunity to store their baby's stem cells away at birth. There's something called cord blood banking. Most of you have heard of it. It's actually a pretty big industry. Globally, it's probably a $5 billion industry. Most parents bought into the concept of doing this because they wanted catastrophic insurance. Okay? They wanted something that in the God-forbidden possibility their child developed a malignancy, that they had a tool readily available to address that malignancy. And you know what? It's been proven over a quarter of a century that it is one of the most useful tools for that unfortunate circumstance. But the truth is, it's a lot more than that. Part of the mission we've had is to find a way to advance the use of those cells in far more than just the horrible, catastrophic things, but to use it to fix more mainstream common disorders, diabetes, cartilage industries, concussion, all that sort of thing. That's required number one, a long history of safety of these products, which we currently have, a more receptive regulatory environment so that we're working within the confines, within the prescribed standards of the government. And we wanted clinicians to fundamentally and intellectually appreciate and adopt this technology. And that's where we are today. What's interesting is that a way of describing it is, as Peter says, is for them to understand that a stem cell is in a lot of ways like a miniature computer. Our partner and friend Craig Venter, as you know, is the guy who first articulated that our biological software resides inside of our DNA. Okay? And if that's true, that software resides inside the nucleus of a cell. And that software is transcribed by the apparatus that resides inside the cytoplasm of a cell. It reads the genes, it encodes, those genes encode for the production of peptides and proteins, and then the surface of the cell acts like an I/O system, like a keyboard. And believe it or not, this model actually works. The cell surface, that membrane, when it sticks to a basement membrane or a surface, actually gets a series of keystrokes that tell that cell what to do. Here's the best example. In bone marrow transplantation, our buddy Dan Kraft knows this well. When you harvest a hematopoietic stem cell from bone marrow or from cord blood and you deliver it intravenously, some of those cells wind up going places like liver. When they get to the liver, they figure out where they are, and they differentiate into liver cells. They don't differentiate into bone cells. They don't differentiate into hair cells. And the reason for that is that they're taking chemical, physical cues from that environment, and they're differentiating appropriately. It's sort of like when you walk in the mall and you look at the map and it says you are there. Same thing happens in biology.
Bob, just in the last couple minutes we've got left, I just want to hit on this: the potential for stem cells to impact longevity and disease. Could you just speak to that for a second?
So, right now, most of us recognize that there are beginning to be approvals using cellular medicine to treat cancer, to treat a variety of autoimmune diseases, degenerative diseases. But one of the richest opportunities is to address the loss in functionality that occurs as we age. That's the spectrum of degenerative, age-related degenerative disorders. One of the things which I uncovered with my colleagues at my company Celgene when we were part of that biopharmaceutical business was that if you look at the bone marrow of an individual, the number of stem cells resident in that organ declined exponentially with age. We actually went and chased this down in multiple organs at Human Longevity when we were doing research with our colleague Craig Venter, and found out that in virtually every compartment of the body, the number of stem cells resident in there declined with age. And if that was true, wouldn't it make sense that since every stem cell thinks it's in a fetus, that you might be able to deliver those cells back to address the problems that occur in an aging individual? So here's the bottom line. As stem cells go from that versatile, undifferentiated form into a mature, specialized cell, they're doing a couple of things. They're shutting off parts of the genome that aren't necessary for that specialized function. Okay? A cholinergic neuron in your brain doesn't have to make hepatic factors. So by silencing that part of the genome, what have you done? You've created a more efficient cell, but you've also created a cell that's confined into that functionality. So if you think about it, the number of cells which wind up being available to remodel and remorph you into a completely perfect copy of yourself begins to decline with age, and those changes represent a decline in the reservoir of cells in their quality and their quantity necessary to remodel you. And so stem cell differentiation appears to echo the process of aging, which to me leaves a perfect opportunity for us to use stem cells to reboot the software in the cellular computer, but also to replace and restore the renovating potential in our body so that we continually to maintain a youthful phenotype. Ken Dykenwald earlier today talked about nothing would be worse than expanding longevity without maintaining the health span. It's our job, I think, to find ways to maximize human performance and maintain a youthful phenotype as we age, we think that stem cells provide that great opportunity. So if stem cells are thought of as being, if aging is thought of as being a stem cell problem, any shift in the balance of those undifferentiated cells to the differentiated specialized cells can be addressed by just giving stem cells as you age. And so we think that recharging the regenerative engine is one of the great potential applications of cellular medicine. And by the way, we're not geniuses here. The public is already demanding this. There is a global network of cellular medicine clinics that are deploying cells derived from newborns to address people's age-related conditions. But it's time for that industry to be corralled and controlled in order to make sure that the products are of the highest quality and the clinical application is at the highest standards.
To tease this, next month, Bob, you're going to be announcing a new company you've been working on. Effectively, it's the culmination of 20 years of your work. And so I don't know if you want to give one sentence without saying much about it, but
I'm always the guy who spills the beans. So here's the bottom line. I've been very fortunate, been part of an organization that was part of the hottest biotech company in the history of the industry. And inside that business, we invested heavily at productizing living cells, turning living cells into basically pharmaceuticals. We're going after the big indications, cancer, autoimmune disease, and so on. But we also see, and Peter and I are behind an effort to now take this to the next level, the version 2.0 of this industry, where these cells are made available to address the more mainstream common diseases that make our life less worth living as we get older. And our objective is, we want you to die young, as old as possible.
Please thank Dr. Bob Hariri.
Thanks.